Okay. So 1 minute. Hello. Can you tell me, student, whether I'm audible and screen is visible? Hello. Okay. So let us start the class. Before going to start the class, so in the last class we have did the biomolecules, right? So today we are going to continue the some parts of the enzymes. Okay. So let us start, and we are solving some of the questions, important questions also. Okay, student? Yeah. So here is the question for you. So can you try to answer me? This is about, so this is about enzyme is incorrect. Okay. So this was asked in the NEET 2013. Option A is enzymes are highly specific. Okay. And option B, enzymes are denatured at the high temperature. Option C, enzymes require optimum pH and temperature for maximum activity. And option D, enzymes are mostly protein, but some are lipids. Okay. So can anyone try to answer this? Okay. So I will run the poll. You can meanwhile you answer me. Okay. Yeah, very good. 1 minute. Yeah. So you can answer me for the question. You can answer me in the chat box, students. So which is the correct answer? So be interactive. Otherwise, how can I know that you're giving the answer? Okay. Yeah. So I need answer from you, which is the incorrect about the enzyme. Try, and you can answer me in the chat box also. Janvi Mandi. Try to answer. Is my slide is moving right? If my slide is moving right, can anyone tell me? Can anyone tell me whether I'm audible and screen is visible here? Can anyone from JNV Mandi can tell me whether it is audible? The screen is sharing, right? Yeah. So okay, answer me for this question. It is very easy question, right? Yes. So when it comes to the enzymes, okay, so here they're asking about which one is about the enzyme is incorrect. Okay. So the correct answer is Which is the correct answer? The correct answer is enzymes are mostly protein, but some are lipids. So this is a wrong according to the enzyme. Okay. So that's why it is the wrong. Okay. Yeah, Manoj, you raised your hand. Tell me. You have any questions, Manoj? Manoj, do you have any questions? Tell me. Manoj Kumar, do you have any questions? You can ask me in chat box, okay? Okay, let us discuss why this answer is correct. Okay, see here, when we are talking about the enzymes, 1 minute. Yeah. Jainvi Udampur. Okay, yeah, Jainvi Udampur, you have raised your hand. Do you have any questions? Okay, so see here. So here they are given that enzymes, right, which is incorrect about the enzymes. Students, so enzymes are very important concept, okay? So see here, enzymes are very or highly specific in nature, right? The particular enzyme is going to convert the particular substrate into the product, right? For example, if you take the maltase, right? If you take the maltase enzyme, it is going to break only the maltase, right? Maltose, right? It is good to break maltose, right? It will break and it will give two molecules of the glucose, right? So that is the function of maltose. Is that is going to give, is that is going to act on the lactose? No, right? Yes. So that's why the enzymes are very specific in nature. The enzyme which break down the which will break down the carbohydrate only. The enzyme which is specific for the protein, it will break the proteins only, right? Yes. So enzymes are highly specific in nature. That is correct, right? Okay. So then coming to the enzymes are denatured at the high temperature. Are denatured, right? Because they are made up of proteins, right? They are made up of proteins. So that's why they're going to be denatured at high temperature, right? What happened at the low temperature, students? Can anyone answer me? What happened at the low temperature? What happened to the enzymes at the low temperature? It is inactive, right? It is inactive. So, yes, this is also correct, right? The enzymes are denatured at high temperature. So then coming to the third one, the enzymes require optimum pH and temperature for maximum activity. Yes, without the optimum pH or without the optimum temperature, the enzyme is not going to work. For example, if you take amylase enzyme, for the amylase enzyme, it requires the pH of 6.7 itself only, right? So with that pH only it is going to work. Otherwise, it is not going to show the optimum activity, right? Yes. So enzymes are required, or we can tell that they are the narrow spectrum, right? So they are act as the narrow. Okay. So this is also correct, right? So third option, the enzymes require optimum pH and temperature for the maximum activity, right? So that's why option D is correct. The enzymes are mostly protein, but some are lipids. No, okay. The enzymes are never be made of the lipids. Okay, either protein or RNA. RNA. So we'll call it as ribozyme, right? So that's why option D is the correct answer. Okay. So let us come to the, so how our enzyme is going to work. Do we know how our enzyme is going to work, student? Let us think. That okay. So there is a one famous theory was given by, or model we can tell, was given by Emil Fischer. Okay, here you can see the picture of the scientist Emil Fischer. Okay. So what the scientist told is, we have a lock and key in our home, right? Lock and key. So if that home key is, if that particular lock is having the particular key, right? So we should have to open the particular lock with the particular key only, right? So otherwise it is not at all possible with the any other key, right? Yes. So here what they have compared with this lock and key is, see here, this lock is there. No, lock is enzyme here. Okay. So lock is enzyme here, and this enzyme is having the site, right, where the key will bind, right? So that site we will call it as the active site. Okay, student, active site. Okay. So this enzyme, so this is substrate, substrate is a key. Okay, here substrate is as a key. Okay, so according to this model, what they have told is, so this based on the mainly the active site. Okay, so active site of enzyme. Okay, this active site of enzyme is rigid. Okay, the active site of the enzyme is rigid. Okay, so particular substrate is having a particular shape that is going to bind to this particular key. Okay, sorry, particular lock. Then only it is going to convert the substrate into the product. Okay, so that was about this lock and key model. Okay, so whenever you take the particular lock, particular key, then only the lock is going to be open, right? So this is how they have explained the lock and key model. Okay, so for example, if you take this is the enzyme, this enzyme is having the active site. Okay, see here, this is a box shape active site, right? So now you can tell me the lock and key model. So this is a one substrate, this is the another substrate. This is the substrate number one, this is the substrate number two. Can you tell me which substrate is going to be bind to this enzyme? Either substrate one or substrate two. Can anyone tell me which substrate is going to be bind? I need answer from a student. It is very easy, right? Tell me which one is going to bind according to the lock and key model. I have told you the active site is rigid, right? Rigid means what? It is made for particular substrate only. It is not going to be change the shape. That is what we call it as a rigid. Can you answer me? I know some of you students are answering, right? Yes, very good. So one, right? The substrate one is going to be bind. Okay. So see here, right? This substrate one is going to be bind here, right? Yes, it is going to bind and it is going to convert into the product, right? So this is how the lock and key model is. Okay. So here you have to remember that the active site of the enzyme is rigid. Okay. So then coming to the another theory has another model was given. So that is the induced fit theory. Okay. Another one is induced fit theory. Induced fit theory. Okay, there are two models. One is the induced fit theory and another one is the lock and key model. Lock and key model is the famous. Okay, so let us think about what is the induced fit theory they are given. Okay, so they are telling that the active site is not rigid. Okay, so here the active site is there, right? Active site, where is the active site is present, students? On the enzyme, right? Active site is not rigid. Okay, it is not at all rigid. Okay, for example, according to the induced fit theory, what they are telling, active site is not rigid. So then whichever the substrate the shape is, it is going to be fit in the log and it is going to be change its shape and then it is going to be fit itself with the log. Okay, fit itself with the active site. Okay, so then how it is going to be converted? Let us see. Okay, 1 minute. Okay, see here how they are telling. So this is the same example we will take. Okay, see here, this is the active site. Okay, so this is the enzyme, so it is having the active site, right? So now, 1 minute. Yeah. Okay. See now, so here the substrate is not specifically like the box shape only. Okay, or we can see here. Okay, see here, this is not perfectly box. Okay, it is like this. Okay, so now what happens? So this is going to bind enzyme active site. Okay, this is going to be bind to the enzyme active site. Then it is going to change the shape. Okay, so after binding to the enzyme, it is going to change the particularly shape. Now it become box, right? After binding to the active site, right? So this is how in the induced fit theory, the substrate is going to be bind to the active site of the enzyme. Okay, so what theory we have learned? Two models we have learned. First one is the lock and key model. In the lock and key model, the active site is rigid. The particular substrate only can bind to the particular enzyme. So here also you can see here, right? The particular substrate, the shape is same, So the enzyme active site is also having the same shape, then it is going to bind particularly to the enzyme active site. In the second, you can see, so this is what I have told you in the induced fit theory. So it is having the substrate is not having the particular structure to bind to the active site. But when it binds to the active site of the enzyme, it is changing the shape according to the enzyme active site. Okay. So this is what we told as induced fit theory. Okay. Here, both the substrate and both the enzyme active site is flexible here. Okay. Where in the induced fit theory. I think you understood. If you are anyone having the doubt means you can ask me. I will repeat once again. Okay. So here you have to remember how the enzyme and how this enzyme is going to convert this product. Okay. So here you can see here. So here in the x-axis I have taken the substrate concentration. In the y-axis I have taken the enzyme activity, how the velocity is going to be, how the substrate is going to be converted into the product. Okay. Velocity of the reaction or rate of the reaction we can tell. Okay. See here. So here we have taken the substrate concentration. For example, here I am increasing the substrate concentration to 4, 8 and 10. Okay. I'm increasing the substrate concentration. Okay. But here you have to think the enzyme concentration is same. Okay. Enzyme concentration is same and substrate concentration we are increasing. Okay. So student, always you have to remember whenever we are doing the enzyme and substrate reaction, enzyme concentration always will be less than the substrate concentration. For example, if you think, so you have taken the, you have to convert the curd into the, sorry, milk into the curd, right? How much inoculum you are going to add? How much inoculum? That old curd, you are going to add the another curd as the inoculum, right? How much you are going to add? You are going to add 1 liter? No, you are going to add only little, right? So like that. So here enzyme we are going to add very little compared to the substrate. Okay. See here how our enzyme is going to convert the substrate. You are increasing the substrate concentration. Okay. The enzyme is converting the substrate into the product. Okay. So you are still increasing the substrate concentration. Enzyme is converting into the substrate. Okay. So it is goes on increasing the substrate concentration. It is increasing the conversion of substrate into the product. Okay. At particular time, what happened? So the substrate concentration is more, and all the enzyme was filled up with the substrate. Okay. They are busy in converting this substrate into the product. Okay. On that time, what happened? It will reaches to the maximum velocity. Okay. It reaches to the maximum. It couldn't able to do or convert more than that what it is did. Okay. It will connect same number of the substrate into the product. Okay. So that is what we will call it as the Vmax. No enzyme is free to convert the other substrate into the product. Okay. So Vmax is the maximum velocity of the enzyme. Okay. Here only we will calculate for any enzyme we are given. So what is the maximum velocity of the enzyme? Okay. So this is how we will calculate the maximum velocity. By goes on increasing the substrate concentration, then we will get the at particular velocity, particular concentration of the substrate, what is the velocity of the enzyme? What is the speed of the enzyme? At what time it is going to convert the substrate into the product? Okay. So see here, then here they are given the Km, right? Km means Michaelis Menten constant. Michaelis Menten constant. Menten constant. Okay. So how we are going to calculate the Michaelis Menten constant? Nothing. So we have got the maximum velocity, right? So that is divided by 2. Okay. So Vmax we have got, right? Vmax, nothing. So Vmax divided by 2. Okay. For example, Vmax we have got the 2, right? So 2, sorry, 6. Okay. 6 divided by 2. How much you will get? 3, right? So now you have to take this 3. Okay. For example, you have to take 1, 2, 3. Okay. Here you have to take 3, and from here to here you have to take this scale and you have to come down here. Okay. So you will get the Km value. Okay. So now you will get the Km value. So where this is the Km value of the enzyme. So here you have to remember. So it is maximum nearly 3.9 something, right? So 3.9 something. Km value is 3.9 something. Okay. So see here. So always the enzyme which is having the Km value, okay, which is having the Km value high means it is having the low affinity for the substrate. Okay. Low affinity for substrate. Okay, student. the all the enzyme, if it is converting the substrate into the product, means for example, if you take the maltase enzyme, okay, or the amylase enzyme, okay, if the amylase is going to be break down the starch, means it must have the low km value, okay, but if it is having the high km value, means it is having low affinity for that substrate, okay, so low affinity. So think here, so if it is having the km value, yes, it is having the high affinity, high high affinity for substrate, right? High affinity for the substrate. So student, here you have to remember, whenever the enzyme is having the high affinity for the substrate, then only it will actively, or it will convert the substrate into the product. Okay, this is important. Okay, you remember this. High Km value means low affinity. So low Km value so we will tell that our enzyme is going to active in only the narrow range of the temperature. Okay. 1 minute. One minute. Okay. So I'm audible, right? I'm audible, right, student? Okay, I will off the video. Okay, so see here. So the temperature of the enzyme, how it is going to be worked. Okay, so the enzymes are generally function in the very narrow temperature. Okay, 1 minute. So the enzymes are generally function in very narrow range of temperature and pH. Both temperature and pH are very important for the enzyme to work actually. Okay, so see here, the enzyme show the highest activity in the particular temperature. That temperature we will call it as the optimum temperature. Okay, the enzyme is going to active only in the particular temperature. For example, if you take 37 degree, okay, so many enzymes in our body is active at the 37-degree temperature. Okay, so 37-degree temperature. Okay, so that's what we will call it as an optimum temperature. On temperature, the enzyme will convert the product correctly. Okay, so then coming to the activity decline above and below the optimum temperature, right? Yes. So whenever we increase the temperature or decrease the temperature, obviously it is made up of the protein. So below the temperature, its activity is very less, and above the temperature, it is going to be denatured, right? It is going to be denatured. Okay. So see here, at low temperature, the enzyme is inactive. So student, do you know? So we'll keep our vegetables and fruits in the fridge, right? Why we'll keep? So it will not going to be ripen, or it is not going to be. It is not going to be. Yes, right. So what it is going to be? Can anyone tell me? Answer me. So what happen if we keep the fruits or vegetables in the low temperature or in the fridge? What will happen? I need answer from you. Can anyone tell me? It is one thing I have told you, it is not going to be ripened so fast, right? Because the enzyme which will be ripened, which is the reason for the ripening, which will result in the ripening, are inactive in this stage, right? Then what will happen? I need answer from you, you answer me. Okay. So at low temperature, the enzyme will be inactive. Okay. So at the high temperature, what will happen? It will be denatured because they are made up of the protein. The tertiary structure is the activity. Okay. Whenever the enzymes, it is in the tertiary structure. Okay. So whenever this tertiary structure was destroyed, the enzyme is going to be denatured. Okay. It is not going to show any enzyme activity. So that is the second thing. Okay. Okay. So see here. So here the graph. So here you have to remember the graph first. Okay. So on the x-axis is the temperature and on the y is the rate of the reaction. So see here, whenever we are increasing the temperature, here temperature, so the enzyme activity is less here, right? It is goes on increasing, right? So now what will happen? So at the optimum temperature, the enzyme activity is very high, right? The enzyme activity is very high. That temperature we'll call it as the optimum temperature. Okay. See here, Above the temperature, what will happen? So enzyme activity becomes decreasing, right? The enzyme activity becomes decreasing because it is going to be denatured. Okay, student? Fine. So let us come to the optimum temperature. Same in the optimum temperature also. Here, whenever increasing the pH, it is going to be increase the reaction at particular pH. At particular pH, the enzyme activity is very high. So that pH, we will call it as the optimum pH, right? So that pH, we will call it as the optimum pH. Okay. So here, this is the optimum pH. So you have to take the line into the center. So center which pH will get. So that is the optimum pH of the enzyme. Okay. So here you have to remember the pH is the, on the x-axis you have to take the pH, on the y-axis you have to take the rate of the reaction. Okay. Yes. So here I have taken the example of effect of some of the enzymes. See here. So where is the pepsin is situated in our body? Can anyone tell me? Where is the pepsin is situated in our body? So what is the main function of the pepsin? Can anyone tell me? Can you recall the digestion chapter in year 11? Yes. So it is helpful in the breakdown of the protein. Very good student. Yes. Right? The breakdown of the protein. It is going to be present in the stomach. What is the pH of the stomach? 2, right? Yes. So pH of the stomach, this thing. So 2, right? HCl is going to be present in the stomach, right? The pH of the HCl is 2, right? Here. The pepsin is active on the 2 pH only. And coming to the amylase, I have told you it is going to be present in saliva, right? What is the pH the active? It is here. It is going to be show the optimum pH, that is the 6.7, right? Here, the lipase. Lipase is active at the alkaline pH, so that is the 12. Okay. So student, they may, in the exam, there is a chances of asking like, so they may give you this graph, okay? They will not give the any pepsin, amylase and the lipase, okay? So they will tell you just, so by seeing this 2, by seeing this 6.7 and by seeing this 12, you have to name the enzyme, okay? So that's why you have to practice this type of the question, okay? Okay. So then coming to the classification of our enzyme. So it is very easy to remember. How you can remember? Here. Do you have any tricks to remember, students? Can anyone tell me? How do you remember this? Oath. Yeah. So many of students are telling this thing only, right? Yes. So that is oath lil, right? Yes. So the classification of the enzyme here, oxidoreductase. Okay. I think you have seen this type of the enzymes in the electron transport chain, right? Succinate dehydrogenase, right? So glutamate dehydrogenase in the nitrogen metabolism, right? Yes. So these enzymes do the reduction and the oxidation. It will take one reactant to the reduced and another reactant to the oxidized. So that is the main function of this oxidoreductase enzyme. I have told you the example of the succinate dehydrogenase and malate dehydrogenase. So these all the things will come under this oxidoreductase enzyme. Okay. So you can take about the transferase. What is the function of the transferase, student? Have you learned in the metabolism only? Aminotransferase enzyme, right? So where amino group is going to be transferred from the glutamic acid to the oxaloacetic acid, converting that into the what? Our oxaloacetic acid, then it is convert that into the, can anyone tell me? Aspartic acid, right? So it is going to be convert that into the aspartic acid, right? So that is the aminotransferase, right? So what it will do, it will transfer the group from one substrate to the another, right? So here you can see there is a one substrate, it is having the G as the group, right? It is going to transfer this to the S dash, right? See here, S dash is having the group now, and the from the S it is going to be removed, right? For example, you can take this thing from the glutamate also. What will happen in the transaminase reaction? Glutamate is having the amino group, right? So from the glutamate plus it is having the oxaloacetate. Oxaloacetate is having the ketone group, right? It is not having the amino. What will happen? So this glutamate will give the oxaloacetate amino group. Then it will convert it into the aspartic acid. Aspartic acid plus, can anyone tell me what it is going to be convert? Plus alpha keto glutaric acid. Glutaric acid, right? Yes. So this is what it will do in the transferase reaction. The aminotransferase is the example. Then coming to the hydrolase, what is the function of the hydrolase? So many breakdown reaction is going to be happen in our body, right? So that is because of the enzyme hydrolase enzyme. So the main difference between the hydrolase and lyase is the hydrolase is using the water. Okay, with the help of the water, it is going to be break down the substrate. See here, the AB is there, right? With the help of the water, it is going to be convert this AOH and the BH. Okay, so that is the main function of the hydrolase. With the help of water, it is going to be break down the student. Fine. So then coming to the lyase. Okay, so what is the lyase? Lyase will also break down the substrate into the product, but without utilizing the water. Okay, so remember here, without utilizing water. So student, here you have to remember, with utilizing water means hydrolase. Okay, without utilizing water means lyases. Okay, yes. So then coming to the isomerases, what is the function of the isomerases? So main function is rearrangement. Okay, so rearrangement. So have you remember, student, in your high school, or yeah, in your high school you remember, right? If you are talking means what the teachers is used to do. If you and your friends is talking means what teachers are used to do? They are used to do the change of the place, right? They will change your friend place into the another place, right? Or they will change your place into the another. The same thing is going to be happen here also. Okay. So the enzyme name is isomerase enzyme. Okay. The isomerase enzyme. What it will do? See here now. Your friend, okay. There are three friends are there, right? A, B, C, right? So these three are together now. Okay. So now what will happen? So this enzyme is going to be changed. See here, this is going to be change its position. So that is A, B, C. Now it is going to be changed into the BCA. Okay. So that is the main function of the isomerase. What is that main function? Rearrangement. Okay. So rearrangement ABC into the BCA. Okay, student. Fine. So let us talk about the ligases. Can you tell me where we have learned the ligases? Have you remembered the biotechnology process, right? Yes. So here ligases we are using to join the nucleic acid, right? Yes. So it will joining function, right? Joining of nucleotide, nucleic acid we can take. Okay. Nucleic acid. It is a function of ligases, right? Here X plus Y is there. This is the utilization of the ATP. So student, here you have to remember that ligases always require ATP. Okay. So ligase enzyme require the ATP for joining the two reactants. Okay. For example, so now I have the two stretch of the DNA. So now I have to join this DNA, right? So for this, I have to use the ligase enzyme, right? So this enzyme require ATP commission. It requires some needs. Okay. If I'm doing the joining work means you have to give me some amount. So that amount is ATP. Okay. So XY it is going to be joined and ATP is converting into the ADP plus PI. Okay. So I think you understood this. Okay. So now let us talk about the inhibition of the enzyme activity. Okay. So for example, student, so why we are using this inhibition of the enzyme activity? For example, you can see in some agricultural fields, some you can take some herbivores, some for insectivores. We have to use some of the chemicals, right? For chemicals, insecticides, we have to use. So on that time, this enzyme inhibition is very important. Okay. So where we are going to inhibit the enzyme activity. For example, some insect is there. Okay. So in that insect, the enzyme is very important for that enzyme insect activity. So what we will do, we will inhibit that enzyme. So by inhibiting that enzyme activity, so that insect is not going to be live. Okay, then it is going to be die. Okay, so this is what we will do in the inhibition of the enzyme activity. Not only in the insect, we will do in the plants also. Okay, so let us talk about this thing. So reduction or stoppage of the activity of enzyme due to the presence of adverse condition or chemicals in the called as inhibition, right? So what happens? So some adverse condition also this is going to be inhibit, right? Here in the adverse condition, and if we add the any chemicals, it is going to be inhibit the enzyme activity. For example, if you take in the optimum temperature also we have studied, right? If we increase the temperature, the enzyme activity is going to be decreased. That is also one type of the inhibition, right? Yes. So, but here the inhibition is based on chemical. Okay. So, student, here there are two types of the inhibition. One is reversible and another one is irreversible. What happen in the reversible means the some chemicals is going to be bind to the active site. So, after some time, what will happen? So, we can remove the substrate. So, remove this inhibitor from the active site. Okay, by adding the more amount of the substrate. Okay, so that is what we will call it as a reversible inhibition. The enzyme active site was inhibited by adding the chemical, something is going to be bind to the active site of the enzyme, then it is not able to bind to the substrate, that is inhibited. But we can reverse this reaction by adding the more number of the substrate. This is going to be removed and our substrate is going to be bind. Okay, so this is what we will call it as a reversible. Okay, so then second thing is irreversible. What do you mean by irreversible? So irreversible means the enzyme active site is going to be binded by the inhibitor. Okay, but we cannot able to remove this remove this inhibitor from the active site. So do you think, student, if the substrate is going to be bind to the active site now? No, not possible, right? Because already inhibitor is binded there, right? For example, if you have a uh, now you are sitting in a chair, right? You are sitting in a bench, right? Before you are coming, your friend come and sit there means you are not able to tell your friend to get up, this is my place, right? So what you will do? You will go to the another bench, right? So the same thing happen here. Okay, so the enzyme active site was binded by the inhibitor now, so the substrate cannot come and bind to the active site of the enzyme. Then if the substrate was not binded means what happens? This substrate is not to the product, right? So this is what happened in the irreversible reaction. Irreversible means it is completely not at all possible to uh, remove this inhibitor from the active site. Okay, so this is what happened in the irreversible. Let us study what they are given. Okay, reversible inhibition. It is a type of inhibition where an active site of the enzyme is blocked. Okay, blocked by the other chemical substances. Once the chemical substances was removed from the active site, enzyme become active. Right? Once I remove this inhibitor means now this enzyme was active. Now it can bind to the substrate. For example, if you see, so this is the active site of the enzyme. So what happened in the reversible inhibition? See here. See, 1 minute. Yeah. So now, so this is a substrate. Okay, this is a substrate. Instead of the substrate, what happened? So this inhibitor went and binded nicely to the substrate site. See here, active site. The inhibitor come and bind. Okay. Now what happened? This substrate is not going to be bind to the active site of the enzyme, right? So now what we will do? We can remove this, okay? So after removing this, now our enzyme is become active. So again, the substrate can come and bind to this, okay? That's why it is called as reversible, okay? So irreversible, what they are given? Irreversible inhibition is a type of the inhibition where active site of the enzyme was permanently damaged, okay? Permanently damaged here, okay? So we can't able to remove the inhibitor from the active site. Okay, so this is what about the irreversible. I think if you understood, if you have any questions, you can ask me, student. Let us come to the competitive inhibition. Student, very, very important. Remember this. Okay, so competitive inhibition, what will happen? So now see here, you have only one chocolate. Okay, so now you have a one chocolate, you have a two of your friends. Okay, so now you this one chocolate you have to give to the two of your friends. So what you will do? There will be a competition will happen, right? So the same thing will happen here also. This is a type of competition which is different. Okay. So let us study now. See here what happened. So this is the enzyme. Okay. This is the enzyme. Okay. So this enzyme substrate is looks same. Okay. Here the enzyme only get confusion, okay, whether it is substrate or it is an inhibitor. Okay, see here how it is looks like. So the substrate is like same only, okay, and inhibitor is also same. It will get confusion now. Okay, the inhibitor also same only. So now what will happen? The enzyme got confusion, so it will bind to the inhibitor. Okay, it will bind to the inhibitor. Okay, so now what will happen? This bind to the inhibitor instead of the instead of the substrate. Okay. So now it is possible to convert the substrate into the product? Not at all possible, right? Yeah. So for this, what we have to do, we have to, here the enzyme is confused, right? Because of the one substrate and the one substrate and the one inhibitor. So only one inhibitor is there, right? Now to reverse this reaction, competitive inhibition is a reversible. What we have to do is, we have to increase the substrate concentration more. What I will do now, I will increase the more substrate concentration now. Okay, see here. Now I'm increasing more substrate concentration. Okay, so more substrate concentration I am increasing. So now what will happen? So now enzyme came to know that, oh my god, it is not my substrate, it is a inhibitor. I have to remove this. Okay, so now what will happen? It will remove the inhibitor and it will bind to the substrate. Okay, so what are the things we have to do in the competitive inhibitor, student? So one thing you have to remember here. So what the thing? So the enzyme, the substrate and the inhibitor is same. Okay, it looks same, but so it is reversible, right? The inhibition is reversible. Whenever we increase the more substrate concentration, this is going to be remove the inhibitor and bind to the substrate. Okay, so this is what in the substrate, sorry, competitive inhibition. Let us see what they are given. Okay, so where the substance similar in the structure, see here, the substance similar in the structure, see here, red color and the black color same in the structure, box shape only. To the substrate competes with the, that substance that is competes with the substrate for the active site. Okay, the both the inhibitor and the substrate will compete for the same active site. The enzyme is having the only one active, both the substrate and both the substrate and the both the inhibitor will compete for the same active site. So now what will happen? But could not undergo chemical reaction, right? Whenever the inhibitor binds, it could not undergo the chemical reaction, right? Yes. So that's why we'll call it as a substrate analog. So student, whenever the inhibitor is similar to the substrate, we'll call it as the substrate analog. Okay, see here. The substance which is similar in the structure to the substrate, okay, that we will call it as substrate analog. Okay. Let us see here. Have you learned about the in the TCA cycle? Have you remember, student, in the TCA cycle you have learned about the succinate dehydrogenase enzyme, right? See here, our succinate dehydrogenase enzyme requires the succinate is a substrate, right? But here the malonate enzyme, so malonate is an inhibitor which looks similar like the succinate. Okay, so whenever this malonate come and bind to the succinate dehydrogenase enzyme, it's not going to convert, okay, substrate into the product. Here succinate dehydrogenase enzyme is inhibited by the malonate. So malonate here is the inhibitor. Okay, inhibitor. Okay, it is similar to the succinate. Can I tell that, student? Can I tell malonate is the analog of the succinate? Can I tell malonate is the analog of the succinate? Can anyone tell me? Malonate is the analog of the succinate. Can anyone tell me? Okay. So this is what about the competitive inhibition. Okay. So let us see next. Allosteric modulator. What is the allosteric modulator? You are observing in the animation here. So this is the active site of the enzyme, right? So this is substrate, right? So see here, in the active, this enzyme it is having the two sites. Okay. So one is the active site, other one is the allosteric site. Allosteric site. Okay. So here see, student, in the allosteric modulator or the non-competitive inhibition, we will tell that. Okay, non-competitive inhibition. So one more thing I have to see here. Okay, in the competitive inhibition, what will happen? So Km of the enzyme is going to be increases. What will happen if the Km was increases means it is decreases the affinity of the enzyme for the substrate, right? Affinity of enzyme it is going to be decreases, right? So let us come to the Vmax. Vmax is not changed. Okay, it is not going to be changed here. Okay, students. So coming to the allosteric modulators, what happened in the allosteric modulators? Km is constant. Km is constant. It is not going to be changed any Km, but it is going to be decrease the Vmax. Okay, maximum velocity of the enzyme is decreases. See here, the enzyme is having the two sides. One is the active site, another is the active site. So this substrate will come and bind to the allosteric site. Okay, so this inhibitor will come and bind to the allosteric site. Okay. So see here, active site where the substrate will bind. Before binding of the enzyme, what will happen, students? Here now, what will happen is this: before binding of the before binding of the substrate to the active site, this inhibitor will come and bind to the enzyme inhibitor site, sorry, allosteric site. So what will happen now? The shape of the enzyme is going to be changed. See here, the shape of the enzyme is going to be changed. The substrate could not able to fit tightly to this active site of the enzyme, right? See here, I'm telling that. See here, so it should be like this, right? It should be like this, right? See here, now it is combined bind. After the binding, the change is shaped, right? It become broader, right? So now the enzyme cannot, the substrate cannot bind to the active site, right? So this is what we will see in the allosteric modulator. Allosteric modulator into the allosteric site, it will change the shape of the enzyme so that active site of the enzyme is going to be changed. The substrate cannot bind or fit to the active site of the enzyme. Okay, fine. So let us talk about the, so this is a very important thing we have learned in the, so 1 minute student. Yes. So this thing we have learned in the simple enzyme and the conjugated enzyme, right? So these are the very important cofactors which is required for the enzyme. What are those things? See here, cofactors. In the cofactor, there will be a two types. Okay. So one is the organic cofactor and another one is the inorganic cofactor. You know the difference between organic and the inorganic, right? Yes. So here the organic cofactor, we will call it as the coenzyme. Okay. Coenzyme. So inorganic, we will call it as the cofactors only. Okay, student. See here. It may be a NAD, NADH, where the reducing enzymes requires this, right? Dehydrogenase enzymes, right? Yes. So these all the things is made up of the protein. Okay. So for example, if you see holo enzyme, what is the component of the holo enzyme, student? One is my as the apo enzyme, apoprotein, right? Apoprotein it must require and coenzyme, right? It must require the apoprotein and the coenzyme. Then it will become complete enzyme, right? Holo means complete enzyme required, right? So that in the conjugated enzyme, the complete enzyme required, the apoprotein part and the coenzyme part. Coenzyme means it is the organic factor, right? It is the organic. It is made up of the vitamins, right? It is made up of the vitamins. If we take holo holo enzyme, it is made up of apoprotein, apoprotein part plus cofactor, cofactor, right? So here the cofactor is the non-organic or the inorganic part, right? 1 minute, I will write. 1 minute, student. Here, so the cofactor here is the inorganic. So for example, you can take the inorganic as a magnesium ion or the manganese ion here. Cofactor may be magnesium Mg2 plus, Mn2 plus, Fe, right? So these all the things will come under the inorganic. Okay. So let us see here. Another example I will tell you. Some enzymes is having the prosthetic group. Okay. Prosthetic group. Okay. So student, when it comes to the organic coenzyme and the cofactor, these are loosely arranged to the apoprotein. Loosely arranged. Remove this. Okay, we can remove this by the any chemical reactions. But the prosthetic group here, this prosthetic group is tightly bound to the enzyme. Okay, tightly bound to the enzyme. Bound to enzyme. Okay, so tightly bounded to the enzyme, it is very difficult to separate this prosthetic group from the protein part. Okay, so for example, if you take the example of the enzyme catalase, catalase, or you can take the enzyme peroxidase. Okay, so these are having the prosthetic group heme, HAEME, heme. Okay, heme as a prosthetic group. It is very difficult to remove this heme group from the protein part of the enzyme. Okay, so it is tightly bound. Okay, so these organic factors and inorganic factor like coenzyme and cofactor are loosely bound. We can remove this. Okay, so this is about the this class, student. So we have completed the biomolecules. If you have any questions, you can ask me. If you have any questions, you can ask me, student. Okay, fine. Then let us solve some questions. I need answer from you in the chat box. Okay, be active. So I'm showing the questions. I need answer from every school. Okay, so please coordinators help with me. So I need answer from every student. Okay, so yeah. So the question is, the structural formula given below belongs to. Can anyone answer me? Which is this structure? Recall which is this structure, student? I need answer from the chat box. Okay. I'll relaunch the poll. Answer me. Which is the correct answer here? I need in the chat box. JNV Udhampur. In the chat box also please answer me. HP, JNV Ropa, JNV Shimla students, and Manoj Kumar. Yeah, Manoj Kumar. Yeah. In the chat box you can give me answer. Yes. So I'm going to end the poll. So very good. Everyone answered to the questions. Very good, students. Yes. So be like this. Okay. Answer me in the chat box and in the poll also. So see here, 33% have chosen the option A and 67% chosen option B. No one chosen option C and option D. So why you are getting the confusion, students? Right? So it is not at all a glucose. Right? How come it will be glucose? Right? Here it is a ribose sugar. Right? 1 minute. 1 minute. 1 minute. So 1 minute. My network there. Yes. The correct answer is glucose, right? Yes. It is the glucose, not the ribose. Yes. Yes. It is the glucose. 1 minute. 1 minute. Give me a minute. Yeah. Yes, student. It is a glucose structure, right? Okay. So let us go to the next question. Yes. So the quaternary structure of human hemoglobin is best described as. Can anyone tell me the quaternary structure of human hemoglobin is best described as? I will run the poll. You can answer me in the poll. So student, ribose sugar is the 5 carbon, right? So here I have given the 6 carbon, right? So that's why I took glucose. Okay. Ribose is the 5 carbon sugar. Okay. So it will show the furanose ring, not the pyranose ring. Okay. We have to know the difference between the furanose ring and the pyranose ring. If it is a 6 carbon means it will show the pyranose. Okay. If it is a 5 carbon means it will show the furanose ring. So like the fructose. Okay. So I'm ending the poll. Yes. So I'm sharing the results with you. Yeah. So option A was given by 33% and option B was no one, option C by 33% and option D by the 33 to 33%. Okay. So now we will see the answer. Okay. See here, what is given? The quaternary structure, right? So our hemoglobin is the quaternary structure, right? See here. So I have told you that it is the 2 alpha and 2 beta, right? It is made up of the 2 alpha and the 2 beta. 2 alpha of the, yes, see here, in option A, they are telling that the dimer of identical subunit. No, right? So here we are having the 2 alpha and the 2 beta. So that's why the option A is wrong, right? Option A is wrong here, right? So that's why 1 minute. 1 minute. Give me a minute. Okay. Sorry, my network is very low. So 2 minutes. Yeah. Yeah. See here, students. See here. So that's why first option is wrong, right? It is not identical, right? So that's why the first option is wrong here, right? So first option is wrong. See here, the dimer of the different subunit. So they are telling that dimer means it is having only the two subunits, right? So it is not made up of the two subunits. It is made up of two different subunits, right? So that's why it is not the dimer of two different subunits. Okay. See here, tetramer, right? So it is having the four polypeptide ring, right? It is made up of the four polypeptide, that is the 2 alpha and 2 beta. See here. So they are telling that tetramer, yes. Here, see here, student. Dimer means it is having the two subunits. It is having the two different and beta, right? No, it is having the four subunits, right? So that's why option B is wrong. See here. In the option C, what they are telling? Tetramer of four different subunits. No, obviously it is a tetramer, but it is having only the alpha and beta subunit, right? So it is having only the alpha and beta subunit, right? So that's why option C is also the wrong, right? Option C is also the wrong. So then which is the correct answer? Correct answer is option D. Okay, student. Correct answer is option D. Do you have any questions here, student? Do you have any questions? See here. Tetramer of four different subunits. No. Here we are having only the alpha and the beta, two different subunits, right? So that's why, see here, tetramer of two different subunits, right? So correct option is option D, right? And the four polypeptide with the two alpha and the two beta, two different subunits. Very good. So see here, you have to answer this. This is from the following is least like to be involved in the stabilizing the three-dimensional folding of the most of the protein. So see here I have given the bonds, right? In this bond, you have to tell me which bond is not involved in the protein formation or the tertiary or the quaternary structure of the protein. Can anyone tell me? Option A is the hydrophobic interaction, option B is the electrostatic interaction, option C is the hydrogen bond, option D is the ester bond. So you can answer me in the poll. I will read on to the poll. Yeah. So you can answer me in the poll. Yeah, fast student. I think this is the last question. This is the last question. There will be the another question is there. Yeah. The another question is there. You can answer me in the, you can do that in the homework. Okay. This is the homework for you. These two questions. Okay. Fine. Thank you so much. If you have any questions, you can ask me. Yes. Wait. I will see this. Okay. Very good. Very good, student. Yes. Ester bond. Where is the ester bond is going to be present? Yes. It is going to be present in the lipids, right? Yes. So that's why ester bond is correct. Okay. Can you answer me for this also, student? I will on the poll. Everyone answered option 2. Very good. I will relaunch the poll. Answer me for this question also. First, this is the correct answer. Pick the incorrect statement with regard to the enzyme action. Okay. So they are telling that malonate is a competitive inhibitor of the succinate dehydrogenase. Yes, that is correct, right? Okay. So addiction of the does not reverse the inhibition of succinate dehydrogenase by the malonate. Is that true? Think. Option C, substrate binds to the enzyme at its active site, where the substrate is going to bind to the enzyme in the active site. Option D, a non-competitive inhibitor binds to the enzyme at a site distinct from that of the binds to the substrate. So that is means the allosteric site. Can anyone answer me? Which is the correct answer? No, student. No, I'm going to end the poll. I'm going to share the result. So many of them has took the answer. Option C, no, that is not a correct answer, right? Yes. So which is the correct answer, student? Now only I told you. So melanate is a competitive inhibitor of the succinate dehydrogen. Yes, it is a Competitive inhibitor, right? Competitive, right? It is having the same structure as the succinate, right? So see here, I have told you. So I have to remove this inhibitor means I have to add the more succinate, right? Here, addition of the lot of the succinate. Succinate is nothing but the substrate, right? Substrate. What they are telling does not reverse the inhibition of the succinate dehydrogenase by the malonate. No, in the competitive inhibition, it can be reversed, right? I've told you it can be reversed, right? Option B is the correct answer, student. Okay, option B is the correct answer. So option C is the wrong. The substrate binds to the enzyme at the active site. Yes, it will bind to the active site only, right? How can it binds to the other site? It will bind to the active site only, right? So in the option D, non-competitive inhibitor binds to the enzyme at the site distinct from which the substrate will bind. Yes, the substrate will bind to the active site, right? Active site where the inhibitor is binding in the non-competitive inhibition. It is binding at the allosteric site. Allosteric site. Okay, student. So that's it for today. If you have any questions, please ask me. So let us meet in the next week. Okay. All the best for your studies. Thank you so much.